Hi, I'm Eve and I work in a lab where we sequence genomes. In 2003, after 10 years, the $3 billion Human Genome Project was completed with all 3.1 billion base pairs read. With its success and advances in technology, people can now pay to have their individual genome sequenced. But why would someone have this done? The main reason is that of personal interest, such as their genome or ancestry. Healthcare providers may also suggest the sequencing of your genome to see if you are at risk of developing particular diseases, but I'm sure the uses will diversify rapidly in the future. The technique we use is called semiconductor sequencing. DNA polymerase is a natural enzyme which makes the well-recognized double-stranded helix structure of DNA. The fundamental principle of the sequencing technique involves DNA polymerase, a template strand, and free nucleotides in the cell, selecting one base at a time. When the enzyme receives a nucleotide which is complementary to the relevant base on the template strand, a reaction occurs. However, if the nucleotide is not complementary, no reaction occurs. The DNA sample, commonly saliva, is placed onto a chip with millions of microscopic whorls. Nucleotides are then passed over the whorls one by one in a known sequence. When a reaction occurs, the proton emitted is received by a detector. If there is more than one of a particular base consecutively, the signal received is stronger owing to more reactions occurring and therefore more protons being released. So overall, the graph shows us the sequence of nucleotides used, and due to the complementary nature of the DNA, we can work out the base sequence of the template strand. This technique all takes place in a relatively tiny amount of time compared with a conventional Sanger method, which involved having to cut up the DNA and perform electrophoresis on it. The result, a process for which a fraction of the cost and time of the Human Genome Project can get your entire genome sequenced. As you are probably aware, genome sequencing is a topic that causes heated debates among scientists, doctors, major genetic companies, and the general public. There is one primary advantage of the genome sequencing, and that is the ability to make an informed decision over having or preparing for a child who has a genetic disorder. For example, in certain Jewish communities, there is a 10 times larger chance of a child being born with Tay-Sachs disease because of common ancestry. Although currently there are methods in place to avoid a child being born with Tay-Sachs, if it does occur, both mental and physical preparations can be put in place before the birth. This enables the baby to have the best quality of life. This is similar for a wide variety of diseases and mutations, and with prenatal sequencing, it enables the parents to choose abortion. Despite the majority of characteristics of humans being a result of several genetic interactions, some people would still attempt to produce a designer baby, selecting its appearance, IQ, and many other characteristics. This is seen by many people as unethical and trying to play God. Another disadvantage is the third-party access to your genome. Although it is illegal at the moment, some people still worry about their DNA or test results being kept private. This could result in genetic discrimination in insurance, employment, and within society. The negative psychological effects of genome sequencing cannot be forgotten, especially if it reveals the risk of a disease later in life. We can now generate huge amounts of data inexpensively. National Human Genome Research Institute's investigations have shown that sequencing costs are plummeting and outstripping the curve of Moore's law, which dictates technology advances at an exponential rate. Researchers at IBM are currently working on a $100 sequencer, a chip that could read bases as DNA fragments flow through the nanometer-wide holes on its surface, perfect for clinical use. So we can imagine to see spectacular advances in our understanding of how the genome works, how disease works, and how they're linked. We know roughly all 24,000 genes, but we do not fully understand introns, and there is evidence to suggest that the majority of variants associated with common diseases are in these regions, so more research is required. It has already led to sequencing companies such as 23andMe claiming to assess a patient's risk of developing health problems. For the future, this means millions of patients will be able to have their genetic makeup laid out to them. Jay Shender, a professor of genetics at the University of Washington, says that in 10 years, sequencing will be routine. One's genome could appear alongside other standard medical information as part of your medical record. Overall, it is clear that since the Human Genome Project, there have been major advances in the field of genetic sequencing. The question is, how far will it go? How quickly will we get there? And would you ever get it done?